So yeah, we can start. So today I'm very pleased to be the host for the AFRICAI Journal Club seminars. I am Hasna Azarwawi. I'm a postdoc at the College of Computing at UM6P and share member of AFRICAI. So AFRICAI have a special interest group that was accepted at MICCAI, and our main goal is to promote research in medical imaging and artificial intelligence for the African researchers, because we believe that we can submit our paper in impactful venues and conferences. I am one of the share members and team members of Africa. We have Fleur, who manage the group. We have Marwan Al Khatib, also Jean Rasser Foupek, and Ismail Kone. And we are the members of the educational program of Africa. So we organized the journal club, a monthly journal club. So it started in March and will be remain until September. So the group is to encourage PhD students, master students, postdoc researchers, or young researchers discussions for impactful or high impact paper that they like to present. So either you can choose one of the paper that you think is impactful, and you can participate and be selected to present it in the journal club. So there will be a prize. So we have two awards of 115 euros for the best journal club presentation, and this prize will be decided upon two criteria. The first one is 50% participant votes. So by the end of the seminar, you will have to vote the quality of the presentation from 0 to 5, meaning that poor, good, average, good, or excellent. And also we'll have 50% evaluation by the educational shares, including me, Marwan, Jean Rasser, Ismail. So today we are very pleased to welcome Ayomide Benjamen Oladele, and sorry if I mispronounced your name. He is a software engineer at the Medical Artificial Intelligence Laboratory, my lab in Lagos, Nigeria, and we'll let him to present himself. He'll do it better than me. And today I'm very happy because he was selected, and he will present us the paper titled Optimized Brain Tumor Segmentation for Resource-Constrained Settings, VGG-Infused Unit Approach. So I leave the floor for Ayomide, and I'll be in the background. And of course, feel free to post your question in the chat box, or post your question directly in the meeting.
All right. Thank you so much, Hasna. Thank you. So quickly, I want to be sure that you can hear me and can also see my screen.
I just stop sharing my screen so that I'll leave the floor for you. I think now you can. Yeah, okay.
Okay, I think everything is good, so we can start. All right, so good morning, afternoon, or evening to everyone, wherever you're joining from. It's a pleasure to be selected by the AFRICAI Journal Club to present today's paper, which was authored by researchers from the Medical Artificial Intelligence Laboratory in Lagos, Nigeria. Once again, thank you for joining this section. My name is Ayomide Oladele, and I'm a software engineer and researcher at my lab, that's Medical Artificial Intelligence Laboratory. And I'll be presenting a recent paper titled Optimized Brain Tumor Segmentation for Resource-Constrained Settings, VGG-Infused Unit Approach, authored by Mizanu et al. And published in 2024 in the Communications in Computer and Information Science Journal. In this presentation, I will walk us through the background and rationale for the study, the methodology employed, and the key findings and a critical appraisal of this paper. We will also have a short discussion at the end, where I look forward to hearing your beautiful, intelligent insight. But before we dive in, I would like to pose a question just to, hopefully this question can stir some reflections at the end of this presentation. The question says, what if we could bring the power of advanced brain tumor segmentation to clinics that do not even have stable electricity? Right. We're going to get back to this question at the end of the presentation to discuss how far the authors went in addressing this, and how far we as researchers can go in pushing the boundaries even further. So let's delve into the paper. Of course, brain tumors. Brain tumors are they are one of the most deadly form of cancer right out there worldwide, reaching about as high as 25%. And in many cases, the difference between survivor and the loss comes down to timely and accurate detection. Unfortunately, in many regions, particularly the sub-Saharan Africa, the odds are stacked against early diagnosis here. So, where we have limited imaging technologies, manual segmentation is the norm. And, the healthcare infrastructure, the fragile, how fragile they are also compounds these challenges. Well, this is where the artificial intelligence come into play, with the potential of improving diagnostic accuracy, reducing workload on already overwhelmed medical professionals, and also ultimately saving lives, especially in resource constraints environment. So now that we have established the context, let's ask ourselves, so why did the authors embark on this research? Some of the challenges presented by the authors cut across, the scarcity of human resources to inadequate infrastructures to, limited funding, insufficient funding for cancer research, even to low availability of computing resources, and also shortage of trained personnel. As a researcher in Africa, I can see firsthand how many of us in this region struggle to deploy machine learning models due to insufficient computational power. And this is why I was also motivated to choose this paper for presentation. Interestingly, so the authors aimed to tackle some of this problem, and their goal was to develop an efficient segmentation model that functions efficiently in low resource settings. More specifically, they set out to reduce the computational complexity of traditional deep learning models, with making sure that they do not compromise on segmentation accuracy. And I believe this is critical because even the best model in the world today, it's going to be useless to us if we can't deploy that model in our environment. So that means for every model we build, it will be nice that we have the ability to use that model in our environment. Now we are going to look into how the authors approached this problem through their methodology. Firstly, the authors made use of 60 adult glioma cases from the sub-Saharan Africa, collected from publicly available BRAS Africa dataset. And a brief knowledge, as we all know in our supervised machine learning models, where you train the model with both the question and the answer. Basically, it's like you have a model, you give the model different images, and you tell the model, this is an image of a dog, this is an image of a cat. And after the model has seen a lot of your information over time, then you just come up one day and say, I have an image for you. Can you tell me what this image is? Is this a cat or a dog? So then the model, based on the previous knowledge and intelligence, can tell you this is a dog. So that's a supervised machine learning model. And it's the unique thing about supervised machine learning model is that they are trained on features and the target variable. So in this case, the features are our brain tumor images, and our target variable is to tell if there's a tumor in this particular image or not. So the paper focused on using brain tumor images that are made up of multimodal scans. So when I say multimodal scans, I am referring to different, types of brain scan images taken with the MRI machine. And we have the T1, we have the T1CE, we have the T2, we have the FLAIR. So in a nutshell, I'm just going to explain what they mean and what it stands for, basically. So the T1 is just an image that shows the structure of the brain. The T1CE, it's an image taken when a particular dye is injected into the machine. So basically, it helps to highlight abnormal areas like tumors. It makes it clearer. And also, the T2 shows more details about, fluid in the brain. This makes it easier to spot swelling or cyst in the brain. And finally, the flare is a type of scan that makes the fluid just darker, as we can see right here. It makes the fluid darker, and it's easier to see brain abnormalities and also swelling or scarring. Together, the authors made use of 4 multimodal scans. So basically, we are saying they used different views of the brain, revealing different details and giving us more insight, or in this case, giving the model more insight. Then the target variable, this case, is the annotated tumor task. So we have 3 categories inside this mask. We have the necrotic core, the enhancing tumor, the peritumoral edema. So the necrotic core is the dead center part of the mask, and the enhanced tumor, that's the active growing part of the tumor that takes up the dye. Then the peritumoral edema is just a swelling, around the tumor caused by the body's reaction. So in a simple term, all I'm saying is that the authors trained the model by providing with images that were already labeled with, this is a tumor or not, this is a necrotic core, this is enhancing tumor, this is peritumoral edema. Then this model, the model they used became capable of analyzing new unlabeled images and also predicting whether or not a tumor is present. So finally, on the data set, they split the data into 2 categories where 997 of this data were used for training and 427 of data were used to validate the model performance. So now we understand the kind of data that was used in this case. Now we go into what they did exactly. So they started by decomposing 3D images to a 2D format. How they did this was they combined the 4 multimodal scans, right? The multimodal scans, they merged them into one single image. So they did that. The idea behind this is they did this so to reduce the computational demand of the model when being trained on this particular data set. And for the mask, recall that I mentioned earlier that the mask, we have 3 categories of the mask: the necrotic core, the peritoneal edema, and also the enhancing tumor. So these masks were split into 3 different categories. So basically, we have a pair of necrotic core and the merged data set, so the feature and the target variable. So we have the necrotic core serving as a target variable. So the model is trained on that. Then the second scenario is the feature and the peritoneal edema. In this case, the peritoneal edema is also the SNFH. And the third case is where we have the enhancing tumor paired with the matched data set. So overall, we have 3 scenarios of segmentation. Case 1, we're trying to segment, is necrotic core present or not? Case 2 is SNFH or peritoneal edema present or not? And case 3 is enhancing tumor present or not? So again, one key thing to pick from this is that they converted the 3D into the 2D data. All right, I've been mentioning models. I've been talking about machine learning models, but now let's look at the kind of models that the authors actually used in their data set. The first one is the standard UNet model, which is popular for medical image segmentation, as you can see here, and it got its name from the structure. You can see that the image of the architecture looks like a U. It's U-shaped. I'll just break things down here to make it look so that we can understand better. So basically, the standard UNet is made up of 2 parts, basically, the encoder structure and the decoder structure. So what that means is that the encoder structure receives an image, right? So when it receives an image, it starts breaking down this image into little chunks, right? So once it's breaking down the image into little chunks, it starts extracting important features from this image. So the important features it extracts from this image, it holds onto it, then starts moving down here, then start climbing up again. So the process of climbing up again is the decoder part of the unit. And this is where it starts reconstructing the image, this time around with focus on the important features it has extracted. But hold on a second. So if the model removed some of the fine details earlier during the encoder section, how does this still rebuild the image accurately since it has removed a chunk out of it, it has removed some details out of it? That's where the skip connection comes in. So the skip connections, I call them the memory, my memory card, right? They're like the memory card. They act like a memory. They keep the original high quality version of the image on the slide. And so when the decoder starts its work, they copy the original image and paste from the encoder part of it and paste it to the decoder. So the decoder is rebuilding and reconstructing the image, having access to the original image. And that's how the skip connections come into play for the unit structure. Basically, some call them skip connections, so you could just call them the copy and paste function. And finally, the standard unit comes with fully convolutional network. What we are saying here is they don't use dense layers, they use only convolutions. The implication of that is the convolutional network helps to, they don't just make one decision from the whole image. They look at every single pixel of the image and they decide if this pixel is a tumor or no tumor. So let's say it's more like you're coloring the image pixel by pixel, you're coloring it and marking the tumor areas in one color and the area areas in another. So this is why, again, the standard unit model is really popular for its ability for efficient image segmentation. The second, I would say, popular model of that the authors also considered is the VGG-Net19, which is the visual geometry group. This is a very powerful model. It's a deep neural network, right? So it's made use of 19-layer architecture. I mean, when you hear that 19-layer architecture, that is deep. That is very deep. And this deep structure allows you to learn complex patterns, excuse me, from the images. You know, it learns the edges, it learns the shape, it learns the texture, and even the object. And the VGG, I call the VGG a camera, a very a sharp camera. So basically, it's like we have a camera from 1999 versus a camera from 2025. You know, when you take pictures with both of these cameras, you can tell that the 2025 camera can show you the image you've just taken, you know, in a cleaner, finer detail. It's sharper, it's, you know, it's better. So basically, the VGG-Net19, you know, goes through the images. The architecture helps it to define edges and textures and shape, like I mentioned earlier. All right, so this is a powerful model, and one special thing about this is it's a transfer learning friendly model. Due to the deep architecture, the model, the authors did not implement this model from scratch. So what they did was that they used this model as a by applying transfer learning. So they used an already trained VGG model, right? So after using an already trained VGG trained model, this already trained VGG model was then inserted into the standard UNet model, as you can see in this picture right here. So you can see this brown arrow here, that's the VGG-Net model. So they still maintain the standard UNet architecture, but this time around, they replaced one of the skip connections and, you know, punched the VGG-19 model into it. So it's more like replacing a 2025, a 1999 camera, you're replacing it with a 2025 camera. So you can imagine how efficient that model is expected to perform. You know, it's basically, I'm saying the authors combine the brain of a specialist, that's the UNet, with the sharp eyes of an image expert, that's the VGG-19, to do a better job. Okay, so now we understand how the models the authors have used, a VGG, a standard UNet model, and also a VGG-infused UNet model. Right now, how they now implemented the model is by training 6 models. How do we have 6 models? I would like you to recall when I mentioned that we have 3 masks: the necrotical, the peritumoral edema, and the enhancing tumor. And they decided to train each model for each of the masks. So we have 3 UNet model for each of the masks as target variable, then also 3 VGG-infused UNet model also on the same data set for each of the categories of the mask. So this makes it 6 models in total. And the models were trained for 200 epochs with a batch size of 64 using AppDam Optimizer as a, to optimize it, and with a learning rate of 0.0001. The model performance were also evaluated, of course, not with accuracy, but with dice coefficient, and the dice loss was also computed. The models were implemented in Keras, TensorFlow, using an NVIDIA Tesla T4 GPU CUDA 11.2. Okay, now we've talked about how the authors handle their models and also how the training was done. Now let's go into the results of this paper. Okay, from this paper, we have two things. We have a pictorial representation and also a tabular representation of the results. From the picture, we can see the authors compared the performance of the standard unit result up here versus the VGG-infused unit result down here. Also, both compared to the ground truth. We can see by close observation, or even if you don't even need to be close. From observing from a far distance, you can see that the VGG-infused unit was able to perform a very close segmentation. The shape of the VGG-infused unit was very similar to the ground truth compared to that of the unit. Let's look at this first column, for example. This is the ground truth. You can see how well-tuned the VGG unit is performing in comparison to the ground truth compared to the unit. So from here, backed up by the tabular representation, we see where the dice coefficient, we have 81.31% for necrotic core, which is better than the 77.64% that the unit showed, and also 93.16% for SNFH, and also it's 9.39%. So the paper showed that VGG-infused unit performed better than the standard unit model. Okay, we have walked through how the authors handle their data and the models they used. In fact, especially the smart combination of their unit and the VGG-19, it's clear that they put a lot of thought into building a system that is both efficient and effective. The way they tackled tumor segmentation with limited data, that's impressive. But beyond the technical process, it is also important to take a step back and look at the bigger picture here. What's worked really well, and also, just as importantly, what could have been better? So from the result presented, we see that the proposed VI unit models clearly stood out. It delivered more detailed segmentation across all the three subregions, and also, they also compared it when compared to the baseline units, and they also achieved higher accuracy, higher dice score, even with a small data set that was used for this training. And the paper also compared this model, the performance of this model with existing framework, and this model also outperformed it. But however, despite this strong result, the paper also presents a few limitations that are worth discussing. First, the model processed 2D slices of the scans. And I believe that this might make the model to lose out on 3D volumetric information, valuable 3D volumetric information that could be useful in understanding the full extent of the tumor. And secondly, they also did not show the deployment or tests of this model in a clinical or real-world setting. So we don't know how the model performed outside of a controlled experiment, right? And lastly, the authors, even though the authors emphasized the use of this model in a low resource settings, they didn't quite provide any data on the model's actual hardware requirements or power consumption. And I believe these are the two factors that are very good in deploying AI in environments with, limited infrastructure. But even with the limitations from this paper, the research laid a big, solid foundation for what could come next for future possibilities. One big step, I think, would be to work with larger sample size. We know for a fact that the more model the data sees, the more data the model sees, the more efficient the model performs. And also, There's also room to get creative with data using smart augmentation techniques like patch-based augmentation, which could help to zoom, on the data set to recognize more detailed regions of the tumor. And yeah, so overall, this step would make the model not just more accurate, but also more ready for the real world. So let's bring everything together now. This study shows that the fusion of VGG-19 and the 2D unit has a lot of potential in pushing the boundaries of medical image segmentation. And through building on the proven architecture and tailoring them to, as a matter of fact, the paper and the authors working with limited data, the authors were able to create a model that doesn't just perform well, but also does this efficiently. I mean, the results speak for themselves. Better segmentation, stronger accuracy, and real promise for use in low resource environment where tools like these are very, very much needed. It's a solid step forward, and it's also a reminder of how thoughtful model design can make a real model, a real impact in healthcare. Okay, so before we move into the discussion again, I will take a moment to thank you all for listening to me and yeah, for listening to me. And also I would like to appreciate the leadership and members of my lab, that's Medical Artificial Intelligence Laboratory, Lagos, Nigeria. Also to appreciate AFRICAI Journal for this privilege. Thank you. I'm gonna show my appreciation for their support, their insight, and also collaboration on this presentation. Thank you so much. So, I don't know, Asne, can I proceed to the discussion section now? You're muted.
Thank you so much, Ayomide, for your presentation.
You're
It was a good presentation. I really enjoyed my time listening to the paper summary. I don't know
thank
if
you.
we should we start? I will start with the attendees' questions, and then I will have some questions for you. So we have question,
Perfect.
Mr. Anis. Uh, what did you use for computing the loss, and what did you propose to mitigate the problem of data imbalance, especially for edema?
So come again with the second part of the question.
So the first one is, what did you use for computing the loss? And what do you propose to mitigate the problem of the imbalance, especially the data imbalance, especially for the edema?
So for data, okay, data imbalance. All right, okay. So
Yeah.
first thing first, I would like to address is that this is not my research. I'm making a presentation on this research. So I did not use, they did. All right. So they used dice loss for computing the loss. Dice loss was used like I mentioned it right here. Yeah, dice loss was used for computing. And regarding the imbalance, the paper didn't mention any imbalance in data set, nor the addressing imbalance in data set again, right? I think I hope I've been able to answer that question, right?
A part of the data pre-processing or the data augmentation of the data.
Come again, sorry.
Can you hear me now? Do you hear me?
Yeah, perfect now.
I'm
Yeah.
on the phone, that's why you may hear some background noises. So I was saying,
Okay.
in the paper, did they present the data pre-processing or data augmentation part to pre-process the images?
Okay. So in the paper, data augmentation wasn't mentioned, and that's why I proposed that when I was doing an analysis of the paper, no, a future possibility of the paper, but the preprocessing was mentioned, and one of the things they did that in fact reduced the computational demand of the model was they converted the 3D form of the data set. We have 3D multimodal scans. So this was merged together into a single image. So that's one of the major key points that I noted from the paper during their data preprocessing.
Okay, I hope that it answers Dr. Annie's question. The second question is, from MRI scan, we get the tree axis of images and how we can integrate the tree of axis to train our model. 997 training data is patient-based or slice-based images. I think you can see the questions in the chat if you cannot hear me.
Okay.
So how did they integrate the three axes of images to train the model? So I think I also
Okay.
quite follow the part where they integrated the three type of images. If you can explain it, please.
We get the three axes of images and how we can integrate the three of axes of training to our. I'm trying to understand the question, but let me address it this way. Okay. So
Maybe
first, okay,
refer
you want to say something?
of the data, the three type of data that you told us that they integrate in your
Oh,
slides of your
oh,
presentation. Mm,
yes. I mentioned that there were three categories of data
mhm,
to train the model, right? So, in machine learning model, we have what we call binary classifications, where you predict a yes or no for a particular data set. So remember, we have three masks. We have necrotic core, we have the enhancing tumor, and we have the peritumoral edema. So what the authors did was that they picked each of this model and they said, let's say, for example, they picked case one where we have the necrotic core. So they predicted, is necrotic core present in this image or not? First category. So a model was used to train and make that segmentation. Then the second was, is enhancing tumor present here or not? Second segmentation. Then the third one is peritumoral edema different? Is it present or not? Third category. So this was done by them splitting the mask into three, like I mentioned here. Right. So it's like we have XY. The X is like, you were saying something?
Each image we have three masks.
For each images, we have three masks. So it's not like in a basket we have three masks. So our image comes to pick one of the masks. Then we train a model to predict and use that mask as a target variable. Then the image comes back to pick the second mask, right? Then we train the model to perform segmentation. Then the image comes to pick the three masks. It's not the regular type of.
Different data sets so we have the same image with three different masks, and for each mask we have a segmentation for one type of I don't know, a necrotic core, enhancing tumor.
Yeah, exactly.
I think now it makes sense why they did 3 unit and 3 fused VGG-19, because I was confused.
Exactly.
Three units and 3 VGG-19. Okay.
Yeah. So that's why we have 3 unit models. So three, the unit model was used to train one. Necrotic core was a target variable. The other trained for peritumoral edema as a target variable. The third one trained for enhancing tumor as a target variable. So the same pattern was repeated for the VGG-infused unit, and that's where we have 6 in total. Then there was a last question that she mentioned.
I have another question about this part. So
Okay.
units, then do we have different outputs for each of each case of each diagnosis, or do they merge the mask by the end?
Come again. Sorry, I didn't get that right.
So we have one image with three masks, and we have
Yes.
VGG-19 fused unit. They trained unit for one of the diagnoses, so 3 times, one for the necrotic core, one for the SNFH, and one for enhancing tumor. So as output, do we have one of the diagnosis, or do we have, if we want the necrotic core, we need to use the unit that was trained on necrotic core, etc., etc. Because he
Oh, okay. I understand.
don't understand.
Yeah.
Okay.
Yeah, I understand your question. Okay, so let's stay a little bit on this and let me try to get more illustrative. So we have, let's say on your computer, let's forget the fact that this is being done in one paper. So let's say you have a computer right there with you. I have a computer right here with me, and I have a computer with one of the attendees here. So on your own computer, you have the matched brain image and your own, I gave, so we have a supervisor, right? So our supervisor gave you one of the masks, necrotic core, to work on. Then our supervisor also gave me the other mask, enhancing tumor. Then for the third person, gave that person peritumoral edema, right? So on your computer, you use the model, you train your data set with the target variable necrotic core. So when your model is making a segmentation, the output you get is necrotic core or not. Are you with me? Then for me, I have the enhancing tumor mask. So I'm training my image based on the enhancing tumor as a target variable. So when the target variable, when I'm done training, the prediction or the segmentation that comes out, the output will be this image. Is there enhancing tumor there or there's not? Then the third person, same way. So what they did, these authors, is they just used the 3 mask for the same model. So we have 3 funnels. So funnel 1 that was trained with necrotic core gives us a segmentation of the necrotic core. Then for funnel 2 gives us a segmentation of the enhancing tumor. Funnel 3 gives us the segmentation of the peritumoral edema. So in the results, these results then are now overlaid together, for the visual represent. No, we're not even overlaid actually. You can see that's why in the results we have 3 classes of the visual representation here. We can see, okay, the VI unit and the unit, this is their own prediction for necrotic core. So this is the model 1, right? Model 2, model 3. But of course, we already established that in the model 1, we have 2 models compared together, the unit and the VGG-infused unit model. Is that better?
Yeah, I think it answers my question. I hope that it answers the Mersibon question. So the second
Yeah.
part of the 997 training data is patient-based or slice-based images. So it's about the data.
Slice-based. It's
Slice.
slice-based images, slice-based, yeah.
We have another question from Sofiq. Did the authors make any mention of measuring computational demand with flops, for example, given they are targeting a resource constraint setting?
No, they didn't. I didn't catch that. And that's why one of my critical analysis of the paper was that they didn't quantify the power usage or the hardware computing resource or, that they use. So the answer is no.
Okay, we have another interesting question from Kabir Mohammed. So thank you for the awesome presentation. So replacing a skip connection layer with a whole VGG-19 architecture will definitely increase the unit parameters, right? So especially if the fully connected layer from the VGG-19 are retrained. If this is the case, training requirements will be increased. Is that the case? And did they mention the inference requirements? And I think I had a similar question. So maybe it's not very clear in their architecture. Maybe we didn't follow with your presentation. But from what you presented, they actually replaced the skip connection with VGG-19. So
Yeah.
how did they? I didn't understand it. Did they use VGG-19 as a feature extractor? Did they how did they do that, and was it necessary? Didn't it doesn't it add more complexity to the architecture, and we
Okay.
have like a resource constraint, and we have to keep to have increase the complexity. So if you can explain that part of the architecture they proposed.
All right, great. Thank you, Mr. Kabil, and also you. That's a very beautiful question, actually. And I remember while I was reading the paper, this was one of the things that jumped out at me, and I'm like, okay, what's going on here exactly? What's the plan here? So what I see from the paper is that first off, let me come from the replacement of the skip connection with the VGG unit. Again, the skip connection.
Can go
You want to
back
say something?
of the proposed.
Yeah.
Yeah.
Okay. All right.
Thank
So again, you can see from this image here that the skip connection, let me quickly talk about what they do. The skip connection, they retain the quality of the original image. Then they copy that quality, then, gives it to the decoder so that the decoder can reconstruct the image, basically. So it's basically, let's assume, obviously, because they train, no need for assumption, because they actually trained a standard unit on the data set, and they compared it with the one the VGG was increased, and we can see that the performance of VGG-Infused was better. So again, what I said was, it's like comparing the 1999 camera with the 2025 camera. The skip connection that does the copy and paste is like the 1999 camera. And the VGG-Infused unit, Net 19, is now serving a function like a skip connection, of copying the image. So it's like the kind of image it copies from the encoder, it's of more quality than that of the skip connection. And that was what resulted into the improved accuracy at the end of the day. But yeah, to the question of increase in parameters, you are very correct. This led to increase in unit parameters. You're right. And I saw this and I was like, okay, what's going on? What is going on? So they did mention that they had to train on more epochs, more parameters, right? Like I said, there wasn't any mention of the time taken for inference and also the computational requirements. So I guess this is a very important limitation of the paper, and it would be nice to have them here to to answer why did you do this, and why didn't you give them all of it. Yeah, so but that's how best I can approach that.
So Dr. Anis asks, did they apply or did they perform an ablation study without the VGG-19 as a skip connection to actually compare the performance units with and without the skip connection?
Okay, did they compare the performance with all the? Yeah, that's why we have the standard unit result here and the VGG-Infused unit. So they did. So the standard unit result was from a unit, a plain naked unit that has nothing integrated into it. It's just the skip connections, the convolutional neural network, and all of that. So the standard units, sorry, the VGG-Infused unit, that's the one that replaced the skip connection with the VGG-Infused. So yes, the question is they did. That's what we can see in this result here. And Mr. Raymond also mentioned, yes, you were very correct. You're very correct. Okay. The VGG, so he's adding something to us here. He said it's pre-trained, correct? And all the layers were frozen. So it's severed as feature extractors and denoising. Thank you so much. Thank you for having that. So I would like to expatiate on what he said. Okay, so when the person mentioned earlier that the addition of VGG-19 to our unit model, is that not going to add more parameters? So again, the idea of transfer learning is that you don't do the work that has been done again. So that's why a transferred model has its parameters frozen. So basically, you are picking from his intelligence. So this intelligence is what is used for the feature extraction. So thank you so much, Mr. Raymond, for shining light on that. Thank you.
Thank you all. I just have some few questions before, because we have some few minutes and we'll end the seminar. So if I can ask you, since you are not a co-author of the paper, if I can ask you to continue this work and your perspective and in your own critical thinking and what will you change or what will you do to extend this paper? And what you want to address?
What would I want to address? Thank you very much. Okay, so this question, coincidentally, I love this question. And why I love it is that coincidentally, again, in my lab right here, where we have a lot of research going on, one of the research we've just done, which I spearheaded, was building a unit model, a lightweight unit model that can give us promising results on a normal central processing unit computer. In fact, CPU. So we've done this, it's successful, and we hope in the nearest future we're going to publish this paper. So what did I do, or what was done in that paper exactly? We made use of data augmentation. That was one of the very first thing I did. There wasn't conversion from 3D to 2D. I used 3D. but this time around, I used a patch-based extraction. Regularly we have a 256 by 256 size of an image. So we reduced it to 128 by 128. In fact, further reduction to 64 by 64 by 64. So the dimensionality, the size of the image was reduced into smaller patches, then augmentation was added onto it. So when this augmentation was added, then we just used a simple naked with less parameter standard units. So this was what we did. And we evaluated also with dice score and also computed the loss with dice loss. So this was just what we did. And we had a promising result. The training it ran for between 40 epochs to 50 epochs, and the inference time was 1 hour, 15 minutes. So yes, so it's not what would I do? I have done it, and I'm happy to actually answer that question that way. Yeah, so that's the approach we took already. So, for better results in the future and more use in limited environments.
Okay, great. Also, I have another question, maybe the last question. So, from what I understood from the title of the paper, the problem they are trying to solve is brain tumor segmentation,
Yeah,
unstable electricity, or under resource constraints means electricity, maybe also computation power. So,
yeah.
in your opinion, how did they address this problem, and did they do it proposing this architecture?
Okay, so when I was presenting the limitations and challenges that the authors considered before taking a step further in this research, I mentioned quite a number of challenges. I mentioned a limited infrastructure, shortage of, personnel, um, lack of data, limited data quality. This paper, of course, did not address all of these problems, right? They didn't address all of this problem. And that's why we have a limitation for them. But one thing that it did address was the limited quality of data, limited quality, high quality data, right? Because the traditional existing machine learning models, when you want to train them on data set, you're probably talking about thousands of brain images. I mean, from developed countries, thousands of brain images. But in this case, they use just 95, if I remember correctly again. Sorry. Yeah, sorry, 60, sorry. 95 was for another research. So they use just 60 glioma cases. And this is impressive. Again, this is one of the things that I feel like the authors, one of the impressive things they did with their architecture. So again, all the problems were not solved, but some of the problems were solved, especially in the case of data sets, right? With limited data set, which just 60, they were able to get this kind of result in this pictorial representation. So yeah, that's one of the very, very key things that they solved from this research.
Okay, thank you so much, Ayomide, for your presentation again, and also all the answers you provided for the attendees. If there is
Pleasure.
question, maybe we can see if there is another question in the chat box or in the attendees. So if not, I think we're good for today. Thank you again, and let's meet for the next seminar.